The nucleotide sequence in DNA determines the nucleotide sequence in messenger RNA and, consequently, the sequence of amino acids in a protein. A mutation in the DNA can result in a change in the amino acid sequence of a protein. One type of mutation that can occur either spontaneously or as the result of a mutagen is the addition of one or more nucleotides during DNA replication. Because translation of a gene begins with a specific codon and proceeds one codon at a time, the addition of an extra nucleotide shifts the codons in the mRNA. This type of mutation is termed a frame shift mutation. A frame shift affects all amino acids incorporated beyond the original site at which the addition occurred. If the new codon generated by the frame shift is a stop codon, the protein synthesized will be shortened and is often non-functional. Another type of frame shift mutation occurs when a nucleotide is deleted during DNA replication. Deletion of a nucleotide in DNA results in a change in the codons in messenger RNA from the point of the deletion and changes in the amino acids inserted into the protein. A mutation occurs by base substitution when an incorrect base is incorporated into DNA. Some base substitutions occur because purines and pyrimidines exist in two structural forms. The most common form results in base pairing between adenine and thymine and between guanine and cytosine. However, the hydrogen atoms can move to form a base with altered hydrogen bonding properties, creating a tautomeric shift. When a tautomeric shift occurs in adenine, the adenine can bond to cytosine. A tautomeric shift in thymine allows it to bond to guanine. This error in DNA replication is passed on to the cell's progeny. The change in a single nucleotide in the DNA results in a change in the corresponding nucleotide in messenger RNA. The change in the codon can result in a different amino acid being incorporated into the protein. Slip-strand mispairing occurs when a nucleotide sequence is present in more than one copy in a chromosome, and the copies on homologous chromosomes pair out of register. A loop is formed in one strand of the DNA as the mismatching occurs. This loop is sometimes excised by the cell's repair enzymes, producing a short deletion. Some chemicals specifically promote deletions and frame shift mutations by stabilizing the loops produced during slip-strand mispairing, thus increasing the time the loops are vulnerable to excision. Many of these deletions start in the middle of a codon, thereby altering the reading frame. These so-called frame shift mutations cause the information in the gene to be read in the wrong 3 base groupings, distorting the genetic message.
UV light and other ionizing irradiations damage DNA molecules by creating thymine dimers between adjacent thymines in the DNA chain. The dimers weaken hydrogen bonding and lead to deformations of the double helix that create subsequent errors in replication resulting in mutations. To protect cells against such damage, DNA photolyase, a light-dependent DNA repair enzyme, eliminates thymine dimers from the DNA strand. The enzyme binds to a thymine dimer, and upon photoactivation, transfers an electron to the dimer, leading to the reformation of two thymines. The electron is subsequently returned to the enzyme after the dimer has been eliminated. Chemical agents can induce the methylation or alkylation of guanine residues in DNA at the oxygen attached to the 6th position of the purine ring. These methylation changes can result in mutations. The enzyme methylguanine DNA methyltransferase eliminates these methylation events and restores the correct structure to guanine in DNA, preventing mutations from occurring. Nucleotide excision repair is an important general process that cells use to correct many different types of DNA damage, including UV-induced thymine dimers. Although present in all eukaryotes and prokaryotes, the molecular mechanism of this system is best understood in prokaryotes. This animation will illustrate the process of thymine dimer repair in E. coli. The nucleotide excision repair process uses a set of proteins called UVRA, UVRB, UVRC, and UVRD. UVR stands for ultraviolet light repair of pyrimidine dimers. But remember that this system is also used to repair other types of DNA damage. In addition, DNA polymerase and DNA ligase are used to complete the repair. The process begins with the loading of a protein trimer onto the DNA double helix. This trimer consists of 2 UVRA molecules and 1 UVRB molecule. This trimer moves along the DNA, scanning for damage that distorts the double helix. When the trimer encounters DNA damage such as a thymine dimer, it stops. The UVRB protein remains at the damaged site, while the 2 UVRA proteins are released. UVRC then binds to the site and makes 2 cuts on the damaged strand. Both cuts are several bases away from the damaged site. UVRD, a helicase, then binds to the site and separates the segment of damaged strand from the rest of the DNA molecule. UVRB, C, and D are then all released from the damaged region. This leaves a gap that is filled with the proper nucleotides by DNA polymerase and then sealed by DNA ligase. The end result is that the damaged DNA has been replaced with the correct bases.
Although rare, incorporation of base pair mismatches during DNA replication does occur. There are DNA repair mechanisms that can fix such mistakes. One such mechanism is the methyl-directed mismatch repair system. In Escherichia coli, this repair system can detect a mismatch shortly after DNA replication and repair the newly synthesized strand, thus preserving the base pairing that was present in the original DNA molecule. The system relies on the fact that methylation of a newly synthesized DNA strand does not occur until several minutes after the strand is made. This provides a window of time during which the newly synthesized daughter strand in a DNA double helix can be distinguished from the parental strand, which is already methylated. A sequence of DNA with only one methylated strand is said to be hemimethylated. Actual recognition of a base pair mismatch is the role of the mut-S protein. Mut-S binds to a mismatch site and forms a complex with another protein, mut-L. The formation of this complex recruits mut-H to the region. Mut-H binds to a hemimethylated site. The mut-S, mut-L complex interacts with mut-H by a DNA looping mechanism. Mut-H then makes a cut specifically in the non-methylated strand. An exonuclease digests the non-methylated strand from the cleavage site to just beyond the mismatch site. This leaves a gap in the daughter strand which is filled by DNA polymerase III and then sealed by DNA ligase. The end result is that the mismatch has been replaced with the correct base pair as indicated by the original DNA strand.
During the first phase of the cell cycle, G1, there is a chemical checkpoint that a cell must pass to begin the process that leads to cell division. The protein products of several tumor suppressor genes act at that checkpoint to control cell division. Cells with mutated forms of the tumor suppressor genes may not be able to stop the cell cycle at the G1 checkpoint, thus allowing them to grow and divide rapidly and form a tumor. One such tumor suppressor gene is the retinoblastoma, RB gene. It is often found to be mutated in a form of cancer in the eye called retinoblastoma, as well as in many other cancers. Normally, the RB protein will bind to a transcription factor called E2F. Transcription factors are proteins that can activate gene expression. The G1 checkpoint is passed when special cell cycle proteins cyclin and CDK work together to free E2F from RB. The released transcription factor then activates genes that start the cell on the path to cell division. Mutated forms of RB may not bind to E2F, allowing E2F to act without control to promote cell division. Another tumor suppressor protein that works at the G1 checkpoint is P53. This protein checks for damaged DNA, acting as a quality control for the cell. If damage is detected, then P53 both activates a DNA repair system and halts the cell cycle at the G1 checkpoint until the damage is repaired. The halt is accomplished by turning on the gene for another protein, P21, that keeps cyclin and CDK from interacting. As long as cyclin and CDK cannot form a complex, the cell cannot pass through the G1 checkpoint. The P53 gene is found to be mutated in a wide variety of cancers. In these cases, P53 fails to turn on P21 and thus does not halt the cell cycle like it should when DNA is damaged. How is growth of cells controlled? Each growing cell binds minute amounts of positive regulatory proteins called growth factors that stimulate cell division. A specific cell surface receptor recognizes each growth factor, its shape fitting that growth factor precisely. Growth factors work by triggering intracellular signaling systems. Binding of a growth factor sets in motion a cascading intracellular signaling pathway, usually involving phosphorylation of proteins, which activates nuclear regulatory proteins that trigger cell division. For example, the retinoblastoma protein, RB, is a molecule that interacts with many key regulatory proteins of the cell cycle, depending on its state of phosphorylation. When RB is dephosphorylated, it binds to and ties up regulatory proteins such as MYC, which is a protein needed for cell proliferation. When phosphorylated, the RB protein releases its captive regulatory protein, freeing it to act and thus promote cell division. Free of RB inhibition, cells begin to produce cyclin-dependent protein kinases, CDK, and cyclins, and proceed through cell division.